just like always, guys, don't forget that if you do happen to come in late, just let me know in the chat whenever you get here. I follow it all day, so I'll definitely get you. All right. Let's talk about what we're up to today. So I usually start the day off by asking if people had any questions from the previous day. So from the homework or from the questions that you were completing yesterday, is there anything that people would like me to review or go over? I feel like that first day is probably not too mind exploding, but let me know if you've got any questions. Some people dropped by the Google Meet yesterday, which was nice. That's that hasn't been common in previous quads, which is I'm glad people are actually using it. That's great. No questions? Okay. All right then. Let's talk about day 2. So one thing I also want to quickly mention is some people emailed me yesterday with problems with the textbook, logging in, being able to actually like pull textbook pages up. If you're having problems with the textbook, please let me know if you're still having problems, and we'll still try and troubleshoot those. While we're troubleshooting them, just remember that the pages for the textbook. Oh, this isn't on screen. Sorry. I meant to have this on the screen in the background while I was talking. There we go. Don't forget that the pages to the textbook are actually here, at least for the first several chapters, and that's just to provide people with a backup, just in case they're having difficulty accessing the online text. So I have it for like almost the first unit, I think. But yeah, let's figure out your textbook problems, because I don't actually have the entire textbook like that. I only have the first little bit. So day 2 is now visible. Good, should be. All right, so we're going to start today with a note. We're going to discuss classification. So, in other words, there are lots of varieties of things out there, and like, how do we put them in groups? How do we classify those things? And we're going to talk about some different methods of classification. And we're also going to talk about how to make a dichotomous key. I don't know if you've ever heard of those before. We'll get to that as well. It's just like a strategy for labeling and for differentiating between different species. I guess we're going to talk about that first. I mentioned in here that note the vocabulary list assignment. We already talked about that yesterday, so feel free to add any new words there at any point to your vocab list. I have a video here that you're going to watch after the note that is on taxonomy, which is putting things into categories, putting them into groups, living things. And so this is it's a little bit of review of what is discussed in the note, plus a little bit of extra information. And then there's a little bit of work from the textbook. So I discussed how to make a dichotomous key, and you guys are going to do a little activity from the text where you make a dichotomous key. And as normal, like if you're running into trouble while you're trying to make your dichotomous key, let me know, and we will figure out those problems together. Okay, so that's for the first little bit. How do I do this here? I'll probably when we go to do this textbook work, I'm still trying to figure out timing on this stuff. I'll give you a time when we're going to come back. This is a little hard. The last time I did this, we were in the classroom, so I could just ask people, "Are you done?" And then we just kept going when people were done. But it's a little bit harder to do now. So I guess I'll set a reasonable time limit, and then we'll come back and do the second part of block 1 here, where we're going to talk about phylogenetic trees. So phylogenetic trees are very similar to cladograms. We haven't, or they are cladograms rather. We haven't talked about cladograms yet, have we? No. So anyway, it's a way of visually showing how closely species are related to each other based on genetic evidence. Okay, so that's going to come up. So we're going to talk about how to make those in a little lesson. I'll be brief, and then there's a detailed lesson here, a step-by-step on how to make your own phylogenetic trees. It's a Khan Academy video. It's actually quite good. And then you're going to read through tutorial, 1 on page 22 and 23 in your textbook, which goes over the procedure again, but in a written format. And they're going to have you actually make a phylogenetic tree and determine some information about how the species are related to each other. Okay, so that's the second half of block 1. And then in block 2, we're going to start talking about the kingdoms of life. Okay, so it's a little bit shorter. All right, so I'm going to get started with the note right now. I'll lost my train of thought. Is this right? Yeah, we finished right there yesterday. Okay. I'm going to take this off here. It's actually fairly warm in my basement today. Okay, so there's a lot of living things. We don't have an exact number because we haven't observed, or at least recorded, every living thing on Earth. But based on the number that we have already discovered and named, and the number of species that we discover every year, we can make a fairly good estimate of the number of living things on Earth. And that number is somewhere around 9 million species. That's a lot. So there's quite a few living things. Now, unfortunately, that number is going down significantly every year. We're in the middle kind of a mass extinction event, probably almost certainly caused by us, which, based on what you learned yesterday in terms of importance of ecological services, should be concerning to you. Hopefully at the end of this course, maybe it will help motivate some behavior change for yourselves, because or for all of us, really, because we're not doing a terribly good job here preserving Earth's biodiversity. But anyway, there's that was in the article you read yesterday too. But there's about 9 million species, and in order to keep track of these from like a biology perspective, we have to categorize them. We have to like give them names, and we have to organize them in some coherent way, so that we can better understand them. Okay, like, in other words, if you want to study animals, you got to know what the animals are first before you can study them. What, for example, like let's say you're a specialist in spiders. What is a spider? Like, what isn't a spider? How many spiders are there? These are like the sort of like the basic questions that you need to know if you want to study spiders. So that system, or that science, I should say, for categorizing living things is called taxonomy. And it's pretty old. They'll talk about it in the video as well. Carl Linnaeus was sort of like the first naturalist that came up with a good system. I think I got his name down here somewhere. Here it is, Carl Linnaeus. Because he did a lot of grouping of organisms into categories. And pre-Linnaeus, we didn't do a really great job of like actually writing things down and categorizing them in a systemic way that made sense. I mean, we categorize things almost the same way that you would have done it in kindergarten, where you said like, that's an animal, and like that's a plant, and then that's it. That's all we did. We just said it's an animal or it's a plant, and then those are the categories. So that's a pretty terrible system, because first of all, there's things that aren't those two things, and there's also it's not helpful. That amount of organization is not really helpful in science. So Linnaeus did a pretty good job of creating a much more complex system here for categorizing things, and the system he developed is called binomial nomenclature, or at least that's one of the systems that he developed. So binomial nomenclature, binomial just means to name, and nomenclature means name. So binomial nomenclature means to name. And by that, and you've heard of this before, it's the idea that you are labeling every organism with a to-name scientific name. Like rat, like the common brown rat is called Ratus ratus. Okay, so that's the basic system. in that system, and I think we're going to come back to it. Yeah, we'll come back to it in a sec how it works, but it's a two-name system. Okay. He also created levels of organization. And the levels of organization at the very top are very broad levels. He started off with kingdoms in his classification system. We've actually added an additional level since then called domain. There are two domains. Well, there are three domains. There are. Hold on. Actually, we're gonna discuss this later. Yeah. I'm not gonna jump ahead here, but this is the system here. So it goes domain, kingdom, phylum, class, order, family, genus, species. And when we talk about the binomial nomenclature, that two-name name, what we're actually talking about is the genus and the species. So when we're talking about ratus, the first ratus is the genus, and the second ratus is the species, specifically in that genus. So I have some examples below here where we're going to talk about this in a little bit more detail. And we'll go through some species and look into how you would classify those specific species. But each species, every species on Earth, is going to have a position at each of these levels. So it'll be found in one of the domains, and in that domain, it will be in this particular kingdom, and in that kingdom, it'll be in this particular phylum, and in that phylum, it'll be in this class, and in that class, it'll be in this order, and in that order, it'll be in this family, and in that family, it'll be in this genus, and in this genus, it'll be that specific species. Okay? So this list, this level of organization list, as a biologist, is something that you need to know. Okay? So you need to know that domain is the most general, and then in order to the most specific, which would be species, it goes domain, kingdom, phylum, class, order, family, genus, species. You may want to create some type of mnemonic to remember this. Domain, kingdom, phylum, class. Oh, I think I was spelling class with a K. That's the German coming out of me. Class, order, family, genus, species. I don't know what kind of acronym you could come up with for that, but you should know the order from most general to most specific. Okay, just as a heads up here. That's something that's on like every first year like zoology, biology exam if you ever go to university. I guarantee you will have to do that again. So anyway, that's from most general to most specific. And then lastly, we're going to just have a quick look at dichotomous keys. That's a tool for helping identify species. So, okay, getting back to what we were talking about here. Taxonomy, which is what we're going to discuss today, is the science of identifying living things. You go outside and this is a muskrat. This is a white-tailed deer. The science of identifying what it is out there that you're looking at is called taxonomy. Okay, and beyond just figuring out what it is, placing it into groups of similar species is also part of taxonomy. Okay, it's about organizing living things into groups. We talked about this idea of species last time and why it's important to know how many species there are, because the idea of biodiversity is important in terms of our human survival, and it's important in terms of understanding the health of our natural systems, of ecosystems. So yes, the taxonomy is important. It's important to know how many things are out there. I already mentioned this idea that Linnaeus in the 1700s created the first system for this. We've updated it quite a bit since the original Linnaeus system, but that being said, the kingdom, phylum, class, order, family, genus, species categories are the same. We have added on to these. So later in the unit, you're going to be choosing your own species to do like a little bit of a deep dive on. And you'll notice that when you look up the taxonomy for your specific species, it doesn't only have these. So there'll be other levels, like for example, in the classes, there may be like a subclass or a superphylum or a superfamily or a subfamily or a supergenus. In other words, they've added additional levels of categorization into this system. And that can get a little bit confusing. This is the basic system, okay? And this is really what I want you to know. You don't really need to know too much about that extra system. So don't worry too much about once you get into that with your own species. You don't need to worry about the extra levels that are in there. It's not really important information unless you're like a specialist in spiders or fungus or something, and then you are going to want to know that additional information about the classification of your particular living thing. But I mean, realistically, as in grade 11 biology, we don't need to know that. So the basic system is fine for us. Okay, so those groupings, those levels, kingdom, phylum, class, order, family, genus, species, are called genera. Okay, so those are the names of the groupings. As I mentioned, the name of a species always goes the genus and then the species. So there may be more than one species in a particular genus. So, for example, in the moose genus, M-U-S, and notice that it's capitalized and italicized. So when you type these, you always italicize the scientific name and you capitalize the genus. Okay, that's the first name in the binomial nomenclature. So it's always italicized and you capitalize the first name. You don't capitalize the species name. And you'll see a bunch of examples of this below. But that's like the standard format. I look for that later, by the way, on your assignment as well. But so in the moose genus, okay, there are it's comprised of rodents. The rodents have a pointed snout. They have small rounded ears. They have a body length, scaly tail, and they have a high breeding rate. So those are the characteristics of all of the species that are in the moose genus. Okay? There are specific mice in the moose genus. So I wrote below here, the house mouse, that's a common house mouse, is called Mus musculus. Okay? Mus musculus is the common house mouse. That is a specific species, this is the species, in this genus. So when you write its species name, its binomial nomenclature, you include the genus and the species together. Mus musculus is the name of the common house mouse. That's its scientific name. Its binomial nomenclature. Okay, you've heard of other binomial nomenclature names for things before. You've heard of E. coli. E. coli is Escherichia coli. Escherichia is the genus of the bacteria, and coli is the specific species name. So, and we'll look at some other examples as well. But there are some in like common use all the time. Like I'm sure you've heard of E. coli before. But oftentimes the first, the genus is short formed with just a letter. So you might see Mus musculus written as M. It's usually capitalized, and then period, and then the species name M. musculus or E. coli, and that's honestly just for brevity. That's just to save space. So, but its full scientific name would include the genus and the species like this. All right, so that's basic. That's basic binomial nomenclature. So looking at these taxonomic ranks here. Okay, so the ranks are the different levels of taxonomy. So you would call something's kingdom, whoops a daisy. Didn't mean to erase that. You would call something's kingdom the rank, like the kingdom is a rank, then it goes phylum is the next rank, then class is the next rank, then order is the next rank. So these are the ranks. And then you get placed into a specific group in each rank. So I'm going to go through some examples of animals and some, well, not necessarily animals, some of these are plants, and how you would place them in the various ranks as you move down. Okay, so let's look at I think I have human in detail over here. So before I get to the human one, I'm going to do let's do some other ones here. Let's look at the kingdom for just the common white onion. Okay, so this is I didn't write white onion here, but that's what this is. It's a white onion like you'd get from the grocery store. Okay, so first of all of these are within a domain. That's the highest rank. Okay, so the highest rank here is the domain, and all of these things here are Eukarya. Did you guys talk about the difference between a eukaryote and a prokaryote in grade 10? This should have been in the grade 10 curriculum. A simple yes or no in the chat would be helpful here. I covered this in my grade 10 class, but it's very brief. Okay, so very briefly then, so I'm not getting all yeses here. So very briefly, a prokaryote is an organism that does not have a nucleus. It does not have membrane-bound organelles. Okay, so these are fairly simple organisms that like don't have a Golgi, and they don't have membrane-bound organelles inside mitochondria, etc. Okay, so within prokaryotes, there are actually two domains. Okay, so the two domains within prokaryotes are Eubacteria and Archaea, or Archaebacteria. It's also written as Archaebacteria. Okay, those are both prokaryotes. Each one of those is also its own kingdom. A little bit confusing. The other domain, so these are the three domains then. The other domain is Eukarya. Eukarya, or eukaryotes, are things that have membrane-bound organelles, like a nucleus. They have Golgi apparatus. They've got basically all the stuff that you learned about cell parts, except for ribosomes. Ribosomes aren't membrane-bound, but all the membrane-bound ones, Eukarya have those. Okay? So this level right here that I've just drawn is the level of domain. There are three domains. These are the three. And all of the ones that we're going to discuss down here are all from the Eukarya domain. The other two groupings, Eubacteria and Archaea, also have one kingdom in each of them called Eubacteria and Archaea. So the kingdom has the same name. And we're going to spend lots of time talking about those as well. But all the examples that I have down here are from the same domain, and that's from the Eukarya domain. Okay, so the highest level here is domain, and there are three domains: Eubacteria, Archaea, and Eukarya. And of the Eukarya domain, okay, we're gonna look at some specific species in here. Okay, some specific genera, groupings within the ranks. Okay, so let's talk about this common onion first. So it is a Eukarya. Okay, it's a eukaryote, and the reason it's a eukaryotic is because it has membrane-bound organelles. It has a nucleus, it has chloroplasts, it has membranes inside. Okay, so that places it in the domain Eukarya. Its kingdom is kingdom Plantae. Okay, so we're going to go down here one at a time. So there's obviously more than one kingdom within Eukarya, but Plantae is one of them. Plantae are organisms that are sessile. They don't move during their lifetime in terms of location. They photosynthesize, so they are autotrophs. They make their own food. I think, wait, is that? Oh, yes. And they have cell walls that are made out of cellulose. Okay, so all of those things are common to Plantae. So that is, that's what places them in that kingdom. Okay? Now, there's obviously lots of different plants. And so within the kingdom of Plantae, there are different phylums. Okay, there's actually a whole bunch of different phylums. But onions specifically are part of the Magnoleophyta phylum. Okay, that's the genera within that rank. Okay, now I'm giving you a lot of extra vocabulary here. So these are genera within each rank. So within that phylum, Magnoleophyta, they are in a specific class. Okay, so there's more than one organism in that phylum. And some of them are in the Lilopsida class. Okay? And of the ones that are in the Lilopsida class, the Lilialis, they are in the order Lilialis. Okay, let me zoom out here a little bit so it's a little bit easier to see what's going on here. They're in the order Lilialis. And in the order Lilialis, they are specifically in the Lilius... Whoa. How do I say that? Liliaceae? Liliaceae. I'm not a botanist. Some of this stuff is not commonly on my tongue. Liliaceae, family. Okay, and in that family, they're in the Allium genus, and in that genus, their specific species name, also sometimes called the specific epithet, is Cepa. Okay. So, and Kappa, by the way, is just a modification of the word Cepa. Okay, it's the same thing. That's actually confusing. I'm going to rewrite that. I'm going to rewrite that because typically they're written identically. So I don't want to confuse anybody here. Okay, so Allium cepa is the common name, is the scientific name for onion, for the white onion. Okay, so at this point, normally I would look in the classroom and see, are people staring at me like I have two heads? But I can't tell. So I'm going to assume people are okay with this. If you're like, I'm not 100% sure what's going on here, I'm going to do a couple more examples. Keep in mind that you don't need to memorize this stuff, okay? So you don't need to know the kingdom or the order, the kingdom, phylum, class, order, family, genus, species for any of these specifically, okay? But you do need to know the order that these ranks exist, and you will be investigating these for particular species, okay? In other words, you should know which one is the most general, which one is the most specific. But I won't ask you to memorize any of this, like specifically for a species. Okay, so let's look at a different organism. Okay, let's look at a fruit fly. You guys may actually even investigate the fruit fly further. There's an opportunity to do a project on it later. I'll not in this unit, in a different unit. But okay, they are also part of domain Eukarya. Okay, and the reason that they fit in there is because they have membrane-bound organelles. Okay, and of the ones that have membrane-bound organelles, they belong to Animalia, that kingdom. And the reason they go in Animalia is because they move. Okay, so they're mobile or motile during their life. Okay, they are heterotrophs, which means they rely on other organisms to get their energy. So they have to eat things to live. Okay. And they're multicellular. Okay, so multicellular, mobile, and heterotrophic makes you an animal. Okay, you go in the animal kingdom. Those are the characteristics that would place you in the animal kingdom. Now, out of all the animals, and there are lots, and we'll talk about all the different groupings within animals later, they fit into a specific phylum of animals called arthropods or arthropodae. That's their phylum. And that has to do with they have a chitin exoskeleton. Oh boy, I don't remember all of the characteristics specifically that make something an arthropod. Chitin exoskeleton. Oh yeah, they have a 3-segmented body. There's others, other characteristics. Sorry, I'm not an arthropod specialist here, but so within the, Animalia, all of the ones that have those characteristics are arthropods. Okay, within the arthropods, there are a bunch of different classes, and the fruit fly is an insect. It belongs to class Insecta. Of all of the insects in class Insecta, they belong to the order Diptera. And then of all of the ones in Diptera, they belong to the family Drosophilidae. And for all of the ones in Drosophilidae, they belong to the genus Drosophila. And their specific species name, this is the common fruit fly, is Melanogaster or Melanogaster. So the full species name, which again would be the genus and the species for this particular organism, would be Drosophila. Whoopsie-daisy. That's not what I wanted to do. Drosophila melanogaster. Okay? Or melanogaster. So that's the species name for the fruit fly. And so you can see how they fit into various different groupings that get more specific as you move down based on their characteristics. Okay, so I haven't been doing a great job of explaining the differences between these groupings, mostly because I don't always know what the specific differences are between these groupings. I'm pretty good with kingdoms and phylums within each of the kingdoms, but then once we start going lower and lower, I don't know every characteristic for every grouping. So I mean, obviously, something for me to investigate here. But it's based on characteristics. So if you look at humans and dogs, for example, as a comparison, well, we're both eukaryotes. Okay, so we both belong to the same domain, obviously. We both have membrane-bound organelles. We're both animals. In this case, we're part of the same animal phylum because we're both chordates. We both have a spinal cord. So that is one of the groupings within Animalia. We are also both mammals, which is a class of chordates, which is class Mammalia. And mammals are hairy. They have mammary glands, so they feed their young. They have live young that feed via mammary glands from the parent. That's where Mammalia comes from, mammals, mammary glands. And there are a couple other characteristics. They're warm-blooded. And anyway, we share that with dogs. But at the next level of classification, okay, the next rank, we belong to a different order. So we are primates, humans are primates, whereas canines, dogs, are carnivora. Okay, and this has to do with partly their nutritional requirements and some other characteristics. Okay, it has to do with some orofacial characteristics of dogs specifically that differ from that of primates, and etc. So there's multiple things here. And then now that we've diverged into different groups, now the groups keep becoming more specific. Okay, obviously humans are not the only primates. There's lots of primates. Chimps are primates, and gorillas are primates. But we belong to a very special group of primates called hominids. That's our family. Interesting to note that we are actually the only living member of our family, the hominids. In the evolution unit, which we do last, we're going to get into a lot more detail talking about what were the other hominids, because there were other members of our family that went extinct. And so we spent a whole day talking about human evolution. That's towards the end of the course, but we'll go into more detail on what happened to the other ones. But anyway, we're hominids. We're the only hominids left. And within that grouping, we belong to the genus Homo, and we are specifically sapiens, and that makes us Homo sapiens sapiens. You may have heard of Homo sapiens sapiens, with that extra sapiens at the end there. We're often referred to as Homo sapiens sapiens, and the reason for that is there may have been more than one subspecies of Homo sapiens. In other words, there were other Homo sapiens that were characteristically different from us, but we could still interbreed and produce fertile offspring, going back to the biological definition of species that we talked about yesterday. So we wouldn't call them separate species, but they were largely characteristically different from humans, and so we kind of split into subspecies. But we are the Homo sapiens sapiens subspecies. Okay, anyway, hopefully people are getting the idea of how this works. So I wanted to talk about now humans in a little bit more detail about why we fit into every particular group in terms of our taxonomy. Okay, so I'm only going to do this with humans, but keep in mind that you could do this with any organism. You could figure out why they fit into that particular group, and you are going to create this exact chart, but you're going to do it with your chosen species that you pick for the final project for this unit. Okay, so just keep in mind, this is basically the exact thing that you're going to do in that project, but you're going to do it with your own species that you choose. So don't pick, you can't pick humans. Okay, so we're part of Eukarya, that's our domain. Because we have membrane-bound organelles. We're animals because we're able to move. We're multicellular. We're heterotrophs. There's actually more characteristics that are important here. I think I took this from the textbook. It doesn't have the complete list. But anyway, these are the important characteristics. So within animals, we're chordates. As mentioned here, we're animals with a backbone. Chordates actually don't all have backbones. This is from the textbook, and that's an error. But it should actually say animals with a spinal cord. Because there are chordates that don't have a backbone. So that's actually not correct. But okay, I'm not going to give the people the textbook too hard of a time here, but it just means animals that have a spinal cord. So we have a spinal cord. We fit in chordates. Of that phylum, we belong to class Mammalia, and I mentioned already here, we have milk glands, we have hair, Those are the main characteristics. So in terms of primates, the reasons we are primates is because we are mammals with grasping fingers. So if you are a mammal and you have grasping fingers, you can grasp stuff with your fingers, that makes you a primate. A lot of mammals are primates. There are several different types of primates. Of those primates, the ones that have relatively flat faces and three-dimensional vision, okay, and three-dimensional vision is the idea that we have eyes at the front of our head. There are a number of primates that have eyes sort of on the sides of their heads, and they don't have three-dimensional vision because they're not allowed. They don't have binocular vision where they can look with both eyes at the same thing at the same time. But if you do have that, that would make you a hominid. Okay, so as I mentioned, we are currently the only hominids. Okay, this is a little bit confusing because gorillas, I would say, have a relatively flat face, although the way that they're using this to judge here, they're saying that that's not flat enough. It's not as flat as the other hominids, so I don't know where they've drawn that barrier exactly. You'd have to ask a primatologist that question. But there's a barrier where gorillas don't make the cut, but humans and all the other previous hominids do make the cut, and there are a number of other hominids that have gone extinct. And within the hominids, we have an upright body position. Okay, we walk on two legs. You don't necessarily have to be bipedal, but the body position is upright, and it's a large brain to body size ratio. And I don't know what the exact ratio is here. They didn't mention it in the textbook, but it is related to brain to body size ratio. So there are some within the hominid group that have very large brain to body size ratios. Humans obviously are one of them, and Neanderthals are another. So they would also fit into this grouping. They are extinct. And then within that, if you're from the genus Homo, and you have a high forehead and thin skull bones, that places you specifically into the sapiens group. And that is one of the reasons why Neanderthals didn't make it in our group. So Neanderthals are in a slightly different grouping from us. We have a different specific epithet. They're in the same genus, but different epithet, different species. And one of the reasons there is that they've distinguished by head bones. So the forehead is considerably different, and the skull is considerably thicker in Neanderthals. So we don't consider them to be the same species. So that is those are the characteristics that we've used to differentiate between the two. So as I'm going through this, you may be noticing that the characteristics that we're using to categorize here are a little bit arbitrary. I obviously wish that this system was a little bit more scientific, since it is the science of classification. But there are some problems with this system. So the original taxonomic system, the way we categorize these things, is largely based on morphology. So I mentioned what morphology was yesterday. Hopefully you put it into your vocabulary list. But morphology is looking at the physical characteristics and behavior of organisms. And sometimes looking at the physical characteristics doesn't actually give you a good view as to what is related to what else. For example, if you look at a whale, which is a mammal, by the way, a lot of people don't know that. They are mammals. They produce milk. They have live offspring. If you look at a mammal and a dolphin, your first instinct would probably be, well, those are pretty closely related together. They are both mammals. They are both water-bound. They have these streamlined bodies. But they are actually not closely related at all. They are very, very distantly related from each other. Whales, I'll use killer whales as an example because different species of whale actually are not necessarily that closely related to each other. But killer whales specifically are more closely related to wolves than they are to dolphins, which doesn't, on its surface, make sense. If you're just looking at morphology, you might say, that doesn't make sense. Based on the way that they look, they've got to be more closely related to dolphins. That is the problem with only using morphology, a morphological-based approach of taxonomy. So traditionally, we only had morphology to go by. That's the old way, right? I mean, we went out in nature and said, this looks like this, and this looks like this, and that was all we had to go on. And we classified stuff based on that. Huge problem with that. Turns out that a lot of things that look similar are not closely related to each other. So more recently, we've started to categorize things based on genetics. So actually looking at their DNA, and I'm just jumping ahead just for 2 seconds here. We're gonna get to this later on today, but I just wanted to mention we're also gonna talk about this. So that the science of using genetics to fix traditional taxonomy and put things into groups that are actually closely related together is called phylogeny, and we're going to get to that a little bit later today. So sometimes that's also known as modern taxonomy. It is using genetic evidence to sort of rejig the original taxonomic nomenclature. But this stuff right here, this taxonomy, is very ingrained. We've been using these morphological-based taxonomical organizations for a long time. And so it's very deep rooted in science, in biology. And so the change is slow. We've already made some significant changes to the original taxonomical system. For example, there used to only be 2 domains. So there was one, Eukarya, all the ones with membrane-bound organelles. And then there was another domain that had all of the things that didn't have membrane-bound organelles. It was called Monera. But we changed that because we realized that within the prokaryotes, there's actually 2 very significantly different groups within the prokaryotes based on genetic evidence. So we're like, okay, well, that actually doesn't make sense. We have to change the taxonomic system. And we do that all the time. But just the changes are slow. So because people are really into these original groupings here, we've been using them for a long time. So anyway, I just wanted to point that out real quick. It's quite problematic to only look at morphology, at physical characteristics when you're doing this. And I also mentioned this idea at the bottom that since the creation of the original taxa, we've added lots of other stuff. Some of that's based on genetic evidence, but some of it's just based on the fact that we are finding new things that don't fit very well into our original categories, and so we need to create a superorder or a subfamily or a subspecies or a superspecies or whatever. Okay, so that's adding in extra levels into the original taxonomic system. It's not quite as clean as this. This looks clean, like very orderly and clean, but realistically, once you start putting all 9 million in there, it's not quite as clean as this. So just as a heads up, I don't want to deceive people here in terms of how clean this system actually is. So dichotomous keys, that sort of brings us to this idea of, okay, there's lots of things out there. When you go out into the field as a scientist, you got to figure out what's what. You know, you want to figure out how many of your red pandas are out there. Well, you better be able to differentiate the red panda from other similar looking species that are out there. And so that's when tools like the dichotomous key come in handy. Okay, so a dichotomous key is a tool that allows you to figure out whether or not something is in a particular group based on a yes or no question. Does it have this or does it not have this? Or it doesn't necessarily have to be yes or no. It's a split. Where you're splitting something into 2 groups. So like, does it have 3 fingers or does it have 2 fingers? Or does it have, is it have hair or does it have no hair? And the decision-making tool is based on morphology. So that's one of the key things here. It's a morphological-based tool, which is usually pretty good in the field. I mean, you can't quickly, as a biologist, you can't quickly genotype something in the field and figure out what its DNA is. So we need something pretty much morphologically based here to determine what is or is not a particular species. Okay, so it's just a tool. A dichotomous key is a tool. So we're going to look at some dichotomous keys, and we're going to make a dichotomous key, and then you guys are going to make a dichotomous key. Just a tool for differentiating. Okay, so for example, this is a dichotomous key for the invertebrates. So invertebrates are a group of animals. Okay, so again, if we were to go like through the levels here, it would go eukaryotes. Let me write this stuff down here. We'd go Eukaryota, and then of the Eukaryota, this would be in W domain. Kingdom would be Animalia, and within Kingdom Animalia, we would be discussing the invertebrate phylum. Okay. So within the invertebrate phylum, so you know that something is an invertebrate, but you want to know what kind of invertebrate it is. Okay? You can go through this decision-making chart to figure out what are you looking at. So first of all, you look at the invertebrates and you say, is the creature I'm looking at asymmetrical or symmetrical? In other words, is there a line of symmetry in the organism? Okay? So if it's asymmetrical, that is, there you couldn't draw a line anywhere on it and find it's symmetrical with the other side. Okay, that would make it a porifera. Okay, that is a type of invertebrate. If it is symmetrical, well, then we need to ask another question to differentiate. Okay, because there's lots of ones that are symmetrical. So is the symmetry? Is it radial? So radial symmetry means that there's a center point. And then there's different, it's basically like pie slices from the center point that are all symmetrical to each other. Okay, so that's radial symmetry. Or is it bilateral symmetry? You guys know what that means. Bilateral symmetry is when you have symmetry like left and right symmetry. Okay, so there's only one grouping that is radially symmetrical, and that is the cnidarians, which include things like jellyfish. Okay, they're radially symmetrical. But if it's not radially symmetrical, if it's bilaterally symmetrical, there's lots of things in that group. So then again, you need to ask another question to differentiate. So in this case, does it have an anus? Okay, that's a valid question to ask in this group. So if it doesn't, then that would make it a platyhelminth. If it does, well, there's a number in that group that do have one. So you need to ask another question, another dichotomy. Okay, a dichotomy is just a decision between two things, a split. So, is it visibly segmented or is it not visibly segmented? Okay, so mollusks don't have any visible segmentation. They are continuous. Okay, but if you have visible segmentation, there are two groups that have visible segments. So then you need to ask one more question, a dichotomy, okay, a decision between two things, dichotomy, to determine which grouping it falls into. Okay, so does it have an exoskeleton or not? If it does, then it fits into the arthropods. If it does not, then it's an annelid. Okay, or a roundworm. That's another word for annelid. So that is an example of a dichotomous key. All right, we'll make one together in a second, but let's look at another class. Okay, so this is for the invertebrate phyla. Let's look at the vertebrate class. Okay, so kingdom, phylum, class. Okay, so this is within phylum. The vertebrate classes that are within Animalia. Okay, it's in the vertebrate phylum of Animalia. That's right. So this would go Eukarya. Oh, jeez. Eukarya, Animalia, vertebrates. No, that's not right. Chordata. I was going to say that doesn't sound right. Chordata. There we go. There. And within the chordata, the vertebrate. Okay? So, like I said, not everything that's a chordate has a backbone. So within the chordates, the vertebrates are a group within the chordates that do have a backbone. That's an important distinction. That was the mistake that was in the textbook earlier. So within the vertebrates, does it have fur? Okay, if it does, you're a mammal. There you go. Easy to classify that. Does it not have fur? Okay, well, you need to ask another question to help determine what group it's in. Does it have feathers? Yes. Birds, aves. I should probably call this its actual name. These are called aves. No feathers? Well, you got to ask another question. Does it fertilize internally or externally? That means, does it lay eggs that are fertilized outside, or is it fertilized within the organism? Okay, so if it has internal fertilization, that's a reptile. Okay, so in other words, its eggs are fertilized before they're laid. Or external fertilization, eggs are laid and then the eggs are fertilized. Okay, within that grouping, you can differentiate one more time here. Does the adult have gills? Yes or no? If the adult does have gills, it's a fish, and if not, it's an amphibian. Okay? Hopefully people are getting the general idea here. It's a yes, no, one way or the other tree of determining what a specific thing is. Now, these are not determining species here. These are determining just groupings, and you can do that too for the higher groupings within a particular rank. Okay, but you can do this all the way down to the species level. So you can say, okay, of all the fish, let's look at the fish that have I don't know how fish are grouped, but let's say have a dorsal fin and don't have a dorsal fin, or whatever. Okay, and you can eventually break these down all the way down to their specific species. Okay, so you could just keep going with these. When you are creating your dichotomous keys, there are two important rules to remember. Okay, one of them is that your genera, those are the groupings that you're choosing. Okay, in other words, like has teeth, doesn't have teeth, that's a genera. It's a grouping based on morphology. Okay, they actually have to be based on morphological things. Okay, things that are identifiable on the organism. It's really important that you don't use something like looks like a something. Okay, looks. Or here, let's say, let's make a don't list. All right. Don't use looks like a. And the reason you don't use that is because not everybody would agree that it looks like a blank. Okay. They have to be objective. All right. Don't use things like big or small. Okay, it has big ears, it has small ears, or it doesn't have small ears. You know, that is so difficult, because what is big? What do you mean? Like, big for what? Like, I've seen people use this and say, well, this frog has, you know, a big eyes, and whereas the other frog doesn't have big eyes. What does that mean? Like, bigger than what? It's so subjective. So if you're going to, instead of using things like this, use things like describe the shape. Okay, instead of saying looks like a something, you can say circular blank. Okay, or angular blank. Okay, or something like that, something that's descriptive instead of saying it looks like blank. And same thing for big and small. Use something like larger than 2 centimeters. Or you could use something like, let's say you're saying it's the tail longer than torso. Okay, something so that it's objective. I can say, yeah, its tail is definitely longer than its torso. If you say the tail is big, that doesn't mean anything. Like, maybe I think it's big, maybe you think it's small. That doesn't. So just try and stay away from anything like that. Try and make it your groupings as objective as possible. Okay, I will say you also have to create one of these keys for your project, and that is like the #1 killer for people that are doing this project. Is they just have the most subjective things. So when I try and use your key to find your species, it doesn't work. I can't figure out where I'm supposed to go. I end up in the wrong spot all the time, and that's just because things were not objective. Okay, so You want to base it only on morphology. Technically, you can use behavior as part of this, although for me, the person who's using your key and I can't observe the behavior, we're only going to be looking at photos, probably don't use behavior then, if we're just looking at photos. However, in real life, these are often include things like behavior. So just like, for example, it'll say makes a sound, whatever, a repeating whistle or something like that, or doesn't do that. And that's obviously not a physical characteristic that you can observe like see, but you can hear it. So anyway, let's try and limit ours just to morphological characteristics that are physical. Okay. So that being said, let's create a key. Okay. I have never done this before online. I normally get you guys to do that. I don't know how to get you to do this now. We're just sitting in a chat room here. How do I do that? Huh. Well, this isn't going to be as interactive as it was previously. Well, you know what? We only have 2 more days. We only have 1 more day after this. Wait, hold on. Is it Thursday? What day is it today? Friday? Today's the last day of this. Yes, it's Friday. Today's the last day of this. So we're actually going to be back at school on Monday, at least for cohort A. So we won't have to worry about this after today. So I'm not going to stress over it too much. Normally we would make this key together, but it's a little bit difficult to do through the chat. So I'm just going to do it. We'll increase the interactivity once we get back into the room. So I need to try and distinguish here. I want to try and create 2 groups, basically, from this one group based on morphology. So I'm going to look at these butterflies and say, how could I break these down into relatively equal groups based on their characteristics? Actually, I could just put this out to the chat. How could I? What's a characteristic I could choose here where I could say, these 3 have it and these 3 don't? Throw it out there. It's a little hard because this isn't great for putting hands up and stuff. Okay, cool. Does it have antennae? Now, I should mention that these probably all have antennae, but I think they got cut off when I scanned this document. But that is good. Based on what we can see here, that definitely is good. So this splits it into 2 groups. I'm gonna color them to make it obvious here. Okay, so I've got 3 here that have obvious antennae, and then I've got 3 here that apparently don't. So great, perfect. First question. So we're gonna start here with all the butterflies. Okay. Now, this isn't all butterflies in existence, but it's all the butterflies in our pool of butterflies here. Okay, cool. That might be one that I will, Sarah, that I'll use later. Okay. So our first dichotomy, our first question is, do they or do they not have antennae? Okay, and then if you go in this direction, it'd be a yes. I'm gonna try and color code this just to make it clearer on here. So yes does, or no does not. Okay, that's our first dichotomy. And so the red group does not. By the way, this color coding that I'm doing, you don't have to do this color coding. I'm doing this for convenience to make it a little easier for you to see. Okay, so red is no antennae, and green is yes antennae. Okay, so now of all of the green ones, those are the ones that do have antennae, we're going to create a second dichotomy to differentiate between those. Okay, so what's a dichotomy for the green ones that we could subdivide the ones with antennae? This is of all the green ones. Okay, tell me more about wing shape, Sarah. What about the wing shape differentiates them? So, Buzzy, when you say long or short body, that's when we're starting to get into the subjective area where what does it mean to be long or what does it mean to be short? You'd have to say something closer to body extends beyond the wings or body does not extend beyond the wings or something like that. But long or short won't work because it's too subjective. Okay, wings point up or down? Okay, sure. So the top wing, the upper wing, the upper wing tip points up or upper wing tip points down. Sure, that's okay. So I would say, so I guess what people are suggesting here is that this is pointing down and this is pointing up. Okay, and then we'll see. I mean, this is essentially, or is that one pointing down? See, now that's where it starts to get a little bit subjective. I don't know if this one over here is pointing down or up. It's kind of in the middle. I'm gonna try and pick another one. I like where that's going, but I think that one's a little too subjective. Okay, body longer than wings. That's definitely that's doable. Okay, so this one right here clearly has a body that extends beyond the wings, and these ones clearly do not. Okay, they don't extend beyond the wings. So that's a good one. That's a very clearly differentiating one. Okay. Okay, so yes it does and no it doesn't. So now I think at this point only one of them does, right? Yeah, only B does. So this is actually the end here for B. Okay, body extends beyond the wings. B is now eliminated. We've added it. Okay, so now left over here, we've got these two, and now we've got to differentiate between these. So how would I differentiate between these where the body doesn't extend past the wings? Okay. Sure. Sure, we could do circular patterns. These are circular patterns, and these ones are more like geometrically shaped patterns. Sure. Sure. Okay, so let's do another dichotomy here. Circular shaped wing patterns. We've got yes, no, and the one that does have it, yes, is F, and the one that does not have it is C. Okay, so we've created a key here now that covers at least the top half here for 3 of them. All right, and you can see how you could do this for a larger group, right? You can just keep breaking things in half and breaking them in half until you've created a dichotomous key that is for all of them, whatever size group you have. So now let's look at all the red ones. How could I differentiate between the ones that don't have antennas here? Okay, points at the bottom of the wings. That one is a little tough because I don't know if I would call these points. How could I say that in a way that would make it more obvious? Okay, how about this? Points extend from the wings longer than the head. Okay, that's pretty differentiating. So yes, it does. No, it doesn't. Oh, I should hold on. I'll keep my color scheme here. Otherwise, it's going to get confusing. Okay, so the one that doesn't, there's only one that doesn't have those points extending longer than the head, and that's D. So we've got a dead end here. And so now, of the other ones that have the longer wing points, that's A and E, how would you differentiate between those? You could say, by the way, Ella, you could say longer than a certain amount of centimeters, but the problem is here, I don't have this on a piece of paper and I can't measure it. But if I did have it on a piece of paper, I could actually measure and use the measurement. Yep, absolutely I could. Okay, now differentiating between A and E here. What's my option? Okay, sure. Striped torso. Okay, so this one's got a very clearly striped pattern here on its torso. The other one doesn't. I like it. Yes. No. I believe that finishes it up now. So stripe pattern, yes, was E. And stripe pattern, no. A. Okay, so this is a complete dichotomous key. If you had never seen this grouping of butterflies before and you went into the woods, this is like a group of North American butterflies. Okay, you could bring this key out with you and you could look at the morphology of the adults of the population and you could determine which butterfly it is you're looking at. Okay, so that's in a simplistic way here, a dichotomous key. Now, here we're basing it completely on morphology. I should mention that typically biologists like to base their genera, the way that they're grouping it, the reasons for the groups, they like to base them usually on evolutionary characteristics. Okay, so going back to this chart up here where we're seeing bilateral and radial, anus, no anus, segments, no segments, those are based actually on evolutionary characteristics for these organisms. So, I mean, obviously, we don't have that data for these butterflies, so that's not an option. But whenever that data is available in real life, we tend to use that data, that evolutionary data, that interacts with the morphology here in order to inform how we're going to create the dichotomous key, just so people know. And the reason that we do that is because then things that are grouped closely together towards the end of the key are actually things that are more closely related to each other. So that's called a phylogenetic tree. So we're going to get to those momentarily here. So a phylogenetic tree is sort of a type of dichotomous key, but it's based on relatedness. Okay, which is what biologists prefer. However, in this particular case, we don't know anything about the genetics of these butterflies. And so this is a great key. And you could take any one of these. For example, I could take D, okay? Let's just say I had never seen these before and I picked D. Okay, and does it have antennae? No. Points extend from wings longer than the head? No. Okay, well then I've identified it as D. Okay, and you should be able to go through and do that with any one of these butterflies and end up at the right location. If you're doing this, testing your key at the end, and you don't end up at the right location, you've made a mistake. Okay, so it is something to check. That's actually how I mark your keys, is that I do that. I go through and I just say, oh, can I get to the right end here with this species? Can I get to the right spot with this species? And it's a good way to check whether or not the key's working. Okay, what happens if I add a new species? Okay, what happens if this guy shows up? What do you do? And by the way, this happens as a biologist all the time. We're constantly finding new species. That's why when I say 9 million, we haven't identified we've maybe identified 6. There's a bunch that we haven't identified. What do you do? Add it. Exactly. You got to add it in. So you follow it through the key and then you just go to the spot where it fits. Yeah. So, all right, let's do it. Has antennae. Okay. So we're going to go, yes, has antennae. It's going to end up in the top group here. Body extends beyond wings. Body extends beyond wings? No. Okay, so the body does not extend beyond the wings. Circular shaped wing patterns? No. So now we're actually ending up here where C is. So what you have to do in this case, and I know you guys can't do this because it's on paper. I'm kind of a cheat code here. I just need to move this over because we need to create an additional dichotomy in order to accommodate this new species that we've just found. So how could I differentiate between C and the new one? Okay, so this is the new one right here, G. And C, oops. Hold on, let me just zoom out so you can see them both at the same time. How can I differentiate here between C and G? Okay, tell me more about wing shape. I need to ask like a yes or no question. This or that. Wing pattern. Ooh, I like it. Separated wings. That's good. Does it have separated wings? Okay, so C does not. Nope. And our newcomer here, G, does. Okay, so all we've done is just expanded our key from the end somewhere. All right, and that's what you do when you find a new species. Now, ideally, if we have already categorized these based on genetic relatedness, like traits that are related to their genetics, related to how closely they are related to each other in real life, then when we add a new species on at the tip, that species is going to be closely related to its neighbor at the tip. Again, that's a phylogenetic key. That's the goal of doing this in actual biology, not so much in this practice key here because we're not able to do that. But that is what we're shooting for as biologists, is to have this be a chart based on relatedness. Everybody sort of kind of get the idea here. I wish I had a way to... Is there like a poll function in this? Does that exist? There should be a poll function. Maybe I'll find a way to incorporate that. There's got to be a way for me to create a poll. I don't really have anything built in yet. Let me investigate that. I like the idea of doing a poll, but I don't. Can you tell me more about that, Gabe? The likes and dislikes of the video? Or which likes and dislikes are you? Can you guys like and dislike a comment that I make? Does that exist? I don't know this platform super well. Like the live stream. Okay. Sure. I guess people can like reverse that and change it. Does it update like live as you do that? I don't know if it does. It doesn't show the dislikes to me though. It only shows likes. I'm gonna investigate this. I want a polling feature. Let me okay. I can only see the likes when I look at my live stream like heads up display here. But okay, actually, hold on a second. You guys can use like little emojis, right? Why not just drop me like a thumbs up if so far you're like, yeah, okay, I get what this is about. Or a thumbs down if you're like, ugh. Alex, is that a thumbs up? Is that what that is? Okay, I'm going to assume that it is. You got this. That's what it said. Okay, all right. Okay, cool. YouTube emoji. I've never seen that one. All right. Okay, these appear to be mostly positive. Okay. Okay. If I saw a bunch of thumbs down, I would start getting concerned at this point. All right. Let's talk about what I'm going to get you to do. Okay? So, I'm going to get you to go watch this video now. Can you guys see this? Hopefully you can see this. This is a quick little review about the idea of taxonomy. Talks a little bit more about Linnaeus and how the taxonomical system was first created. Once you're done that, on page 32 and 33 in your text, it's investigation 1.2.1. Monkey with hammer. Jacob, I don't know what that means. I can show it to the screen here. I don't have a PDF for this one. It looks like this. So, this is an opportunity for you to create your own dichotomous key. Okay? Please note that you're doing part A. Part B, only number 6. Yeah, sorry about the lag. I have the latency set to like ultra low on my end, which is really tying my internet up here. But it's basically as low latency as I can make it. Yeah, but I realize everybody's got different internet, right? So this is not going to be perfect. Well, that's all right. We're doing our best. Well, I found that when I did this as a Google Meet, when we used to do Google Meet, it wasn't particularly better. There was then we had all kinds of other problems. So anyway, sorry. On page 32 and 33, you're going to do part A, okay, which is you're going to be creating a dichotomous key for sharks, okay, based on their morphology. And then for part B, please make sure that you're only doing number 6, okay. You don't need to do part 5. You don't need to do number 7. Only number 6, and then analysis A through C. So there's a bunch of other analysis questions. Don't do the other ones. They're not important for our purposes, okay? So it's part A, that's you're going to do a shark dichotomous key. Part B, you're going to be creating a dichotomous key of insects, and then which is number 6, and then A through C, you're just basically going to be looking at your dichotomous keys and answering some questions about the keys that you've just made. Okay, so that's the first part, okay, of the homework questions. And the second part here is on page 20. Numbers 5 and 7. Okay? And yeah, numbers 5 and 7. And those questions are all based on the taxonomical system: kingdom, phylum, class, order, family, genus, species. Okay, keeping in mind that as you go down through the list, the further down you go, the more things in common species are going to have. So the more groupings that a species shares with another species, the more things they will have in common with that species. Okay, so that's the second part. I think that for this, we're going to go all the way until 10:20. You're going to need all that time for sure. And then we'll come back after the break and we'll discuss the phylogeny part, the second part here. Okay, it's mostly just this video. There's a very short lesson and this video. If you want to go ahead and watch the video if you finish early, that's fine. But you don't need to do that. So I will see you guys after the break. In this case, it'll be at 11:15 because, or unless you want to do the meditation, in which case we're going to start at 11:05. So 11:15 for attendance. I'll be here the entire time, so if you need anything, just post it in the chat. And we can jump into a Google Meet if you want, if I need to sketch something out for you or you want to talk about a specific question. Okay? All right, guys. Let me know what you need, and I'll give you a chance to get started on that now. Hey, guys. Welcome back. Some of you, hopefully, are interested in doing the meditation. If not, that's okay. No problem. I'm going to start it right now. Give me 2 shakes here. And if not, cool. I will see you in a few minutes. Quarter after. Just gotta find the right one here. Guided. Here we go.
So as you just take a moment or two to get comfortable, just setting up the exercise with the eyes open. Without moving the eyes, make sure you can see the space around you, either side, above, below. And just maintaining a very soft focus. Just beginning with some big deep breaths, breathing in through the nose and out through the mouth. And as you breathe out this time, just closing the eyes, allowing the breath to return to its natural rhythm. Beginning to feel the weight of the body pressing down, and different points of contact. Just settling into that space. And beginning to notice different sounds around you, just allowing those sounds to come and go. And bringing the attention back to the body now. Noticing any sense of heaviness or lightness in the body. Noticing if the body feels at all restless or perhaps particularly still right now. And then starting at the top of the head, just scanning down through the body, all the way down from head to toe. And as you become more aware of the body in general, starting to notice the breath, the movement of breath. Begin with just in a very general way, noticing which parts of the body move, that rising and falling sensation. And continuing to follow that movement, beginning to notice each and every breath, noticing if the breath's long or short, following each in-breath and each out-breath. And as you continue to follow each breath, just to help maintain that focus, just counting the breaths as they pass, realizing when the mind's wandered, each time just returning to that sensation. So as soon as you realize the mind's wandered, just gently coming back to the breath each time. And now letting go of that focus, just for a moment, allowing the mind to be free. And as you bring the attention back to the body now, just coming back to that feeling of weight, of contact, coming back to the space around you, the sounds, the smells, just becoming more familiar. That space, the way you feel, posture. And then when you're ready, in your own time, you can just gently open the eyes again.
Hey guys, if you joined us for the meditation, hopefully that was restful for you. And if not, that's cool too. If you wouldn't mind just letting me know in the chat if you're here for the second half, and I'll check you off. All right. Let's see if we're missing anyone here. Jacob Ashman. Oh, there's Gabe. Jacob Ashman, you here? What about Alex Puncher? Nope. Emma Rochelle. Alex's wife, I know. Okay, cool. All right, well, she can join us later. Okay, as usual, if you're coming in late, no big deal, just let me know. Okay, so before we move on, does anybody have any questions from any of the stuff from the first block? From the, oh, there's Jacob. From the Dichotomous Key activity. Or other questions from the text, page 20, 5 and 7. It's okay if you haven't gotten there yet. So during learning block 3, that's also an opportunity to finish up the activities. And there's always time for questions at the beginning of Monday as well. Okay. So let's talk about phylogeny. I'm going to head back to the note for a minute here. So, Lauren, you don't submit your homework daily. You keep one Google Doc per unit for your homework, where you keep it. And then at the end of term 1, which is halfway through this quad, I collect the homework as like sort of like a homework check for the quad. It's once as a portfolio. So you submit it once then, and then there's another submission at the end of the course. That's like your homework check for the second half. But it's not, you don't submit it daily. So it's just like stays in your homework log, basically, in your Google Doc that you just keep your daily homework in. Okay. So you don't, there's no place to submit it right now. There will be, like, as part of a portfolio, it'll pop up as an assignment, and we'll talk about it at that point. But you hold on to that Google Doc. Okay, so I mentioned this earlier when we were talking about taxonomy, which is that the idea that just looking at morphology is sometimes a terrible way to organize living things, because they might look similar, they might have similar morphology, but they actually aren't related to each other closely at all. So phylogeny is an attempt to fix that problem, which is organizing things into groups based on their relatedness. And so modern taxonomy is a hybrid of the two, because we don't have a complete set of genetic information for every living thing. And so we're not able to look at the genetics that carefully to organize everything via genetics. However, we use the genetic information to inform our decision making. And when I say genetics, the reason why we can do that is because everything is related. And we'll get into this a lot deeper in the evolution unit. But if you look at DNA between species, there are parts of their genetic code that are the same. So if you look at my genetic code and your genetic code, they were almost completely the same. They were 99.9999% exactly the same as humans. There's a small difference between you and I in our genetic code, but it's quite small. It's really not that many base pairs difference. But if you were to look at like the difference in genes between you and a dog, there still is a lot that's the same. As mammals, we probably share 96% of our genome with other mammals. 97%. A really high percentage. So those sections that are the same are called homologs. They are areas of your genetic code that code for the same things, basically. I mean, when you think about the actual physiology of us and dogs, think about how similar that physiology is. There is. I'm so sorry. It's not on the screen. Let me fix that. There we go. Sorry. So all I did here was just highlight homologs. There are areas that are the same. And that is because they code for the same proteins. Okay, so for example, you need to make insulin to help regulate your blood sugar. Dogs need to make insulin to help regulate their blood sugar. You have a number of proteins on the surface of your cells that are used to communicate with other cells. Dogs have almost exactly the same proteins that they use to communicate with other cells. I mean, all the same organs are there, so you need all the information to construct those organs. the construction of the organism in general is very similar. I mean, if you really looked at all the different, even at the molecular level, the similarities between us and dogs, I mean, it's almost all the same. It's very, very similar. So those areas of your DNA that code for building those things are the same code. Those are called homologs. Okay? And the reason that they're the same is because we have a shared evolutionary origin. That is, we have a common ancestor with dogs at some point in our evolutionary history. I'm not going to get into too much detail on how that works at this point, since we're going to talk about it more in the genetics unit. But at least for this point in the course, you need to know that we have a shared genetic history with everything, with all living things. Even if you look at the similarities between us and a mushroom, okay? I mean, at first glance, you'd say, okay, well, nothing is the same. But except on a molecular level, that's not true at all. So mushrooms and us are both heterotrophs, which means that we need to eat things in order to survive. A lot of the cellular proteins are the same. We both have mitochondria. Also all the proteins that operate inside of mitochondria are the same. A lot of the cell signaling proteins inside of the cells are the same. We have the same cell parts, like organelles. And so construction of those organelles is the same. In many ways, as multicellular organisms, the proteins that we use to communicate between cells and stuff is the same. And so there actually is a lot of shared DNA between us and fungus. And those are called homologs. And so, but the further you get from being related to something, the less homologs you have. So obviously, we don't share the same amount of homologs with mushrooms as we do with dogs. So dogs we might share 97% of our DNA, and fungus maybe we share 40% of our DNA. And even if we start talking about organisms like bacteria, which are pretty much as different from us as you can possibly get. They're single-celled organisms. They don't have any cell organelles that are membrane-bound anyway. There are still homologs. There are still sections of their DNA that are the same as ours. The DNA that's used to construct ribosomes, the DNA that is used, or even just the structure itself of DNA. They both have DNA. But there's going to be homologs no matter what. Now, obviously, we don't have a lot of homologs with bacteria, but we do have some. And the reason for that, again, is because we have a common ancestry. So we can look at that, the amount of homologs, to decide how closely we are related to another organism on Earth. If you look at other primates, like chimps or bonobos, those are our closest related organisms, we share almost our entire genetic code with them. They are 99-point-something% the same as humans in terms of their genetics. And so we are very, very closely related. Our common ancestor was not very long ago in time. Whereas our common ancestor with a fish may have been 100 billion years ago. Well, probably more than that. And so we don't have as many homologs with fish. And so and the number of homologs we have with plants is even less, and we split with plants. Our common ancestor with plants is even earlier than that. So anyway, that's the idea. Now, it's hard to imagine that. Seems like a crazy idea on its surface, but you have to keep in mind how long life has actually been around on Earth. Around 3.8 billion years. That's a really, really long time. It's actually incomprehensibly long. And humans as a species have only been around for a very, very tiny, tiny fraction of that. 1/1,000,000th of that. And so there's been a lot of time on Earth for evolution to happen. I'm not going to get into the specifics of how it happened, since that's an entire separate unit. But anyway, speciation, where one species becomes two, has been occurring on Earth for a very long time. Billions of years. And we have common ancestry. Okay, so based on this genetic evidence, we can build a tree of life. All right, that's a tree of life, also known as a phylogenetic tree, is a tree that is built on how we are related to other living things. So if you go here's the tree right here. This is a simplified version of the tree. And you'll notice across the top, they're color-coded here, B, C, and D. Those are the 3 domains, okay? And those are the highest groupings of living things. And so I mentioned that the prokaryotes, the ones that do not have membrane-bound organelles, have their own 2 domains. They have domain bacteria, which includes all these different types of bacteria. There's 1 kingdom in that domain. And domain archaea. And it contains all of a whole bunch of groups of different types of archaea. And they have their own kingdom as well. Archaea, it's 1 kingdom. And whereas Eukarya, this is the 3rd domain, that includes a lot of the living things that you would be familiar with, all of the eukaryotes. So fungus, plants, protists. You're probably not that familiar with protists, but protists, animals, etc. Okay? And based on genetic relatedness, we can figure out roughly how closely related these things are to each other. And you'll notice that there's a big split actually between bacteria and archaea that happened very early on, which is why they get their own separate domains. They're actually quite distantly related from each other, and so it doesn't make sense to keep them in one single group. I mentioned this earlier. I just noted it below here as well, that eukaryotes are organisms that have membrane-bound organelles. I mentioned this. We were talking in the other note as well here, but that's like mitochondria, Golgi, ER, vacuoles, lysosomes, nucleus, etc. And prokaryotes don't have those, and so the other 2 over there are prokaryotes. Those are known as the Monera, the other 2 groupings. And if you trace them way, way back, right to the very beginning, to where we all had a common ancestor, then we get to what I would call the beginning of life. Okay, so this is like the first living organism, which in biology we call the origin. What was the origin like? We don't really know. I mean, we know a little bit about it because we have homologs, common pieces of DNA, that we share with all living things. There's a good chance that we got that common DNA from the origin, the original life on Earth. But what exactly it looked like, no idea. This is too long ago. There's no fossil record for single-celled organisms, really. Certainly not from that time period. So we don't really know a ton about it. We know a little bit about the conditions that were on Earth at the time and how we potentially could have got a species, a living organism from non-living matter, which is what would have been on Earth in the early days. But that's our shared common ancestry. Okay, so we can build this tree of life based on how things are related to each other, and that is called a phylogenetic tree. So this is another way of talking about this. You could say that this is the tree of life, the phylogenetic tree of life. So I mentioned the 3 domains right here. How do we broadly categorize these? These are the biggest classifications, the broadest classifications. So bacteria is one of them, as I mentioned. They are single-celled. They're all prokaryotes. They have no membrane-bound organelles. We're going to talk about bacteria in more detail. We're going to spend a whole day on bacteria next week. Archaea are single-celled as well. They're also all prokaryotes. And they differ from bacteria in that they have a different style of cell wall, which we're going to talk about more later. And they all have sort of a unique hazardous environment survival strategy. So we're going to talk about some examples of some archaea, but they live in all kinds of cool places: volcanoes, Arctic ice, hot springs, places where you wouldn't find bacteria because bacteria will be obliterated by those environments. So archaea are kind of special survivors. And then domain Eukarya, that's the last one here. They do have membrane-bound organelles, so that's all the other ones. Probably the ones that you think of the most when you think of living things. I haven't frozen. I'm just being very still. Giving you guys a chance to write this stuff down furiously. Keep in mind that there are copies of this in the course notes folder that is linked on Brightspace, so you can access this at any time. How are we doing for time? 11:35. Okay, I better keep going here. Okay, so as mentioned here, modern taxonomy favors genetic relatedness. That's how we like to classify things if we can help it. So we still use the morphological approach. We just don't use it as much, or at least we try and avoid it because it's just not as accurate. Which means that this is sort of a work in progress. We change how things are categorized over time. So down below here you will see a cladogram of primates. A cladogram is where species are organized in a phylogenetic tree based on clades. So a clade is a taxonomic group that includes a single ancestor and all of the descendants of that ancestor. Okay, so each clade in a phylogenetic tree is considered a branch on the tree of life. And the tips of the tree represent single species. Okay, if you move backwards on the tree, what you're doing is you're moving backwards in time. So if you can see on the side here, there's actually times listed here from when we split off from our close neighbors here. Okay, so let's look at some examples. So first of all, if you're closer together on the cladogram along the top here, it means that you are more closely related. This is purely based on genetic relatedness and homologs, how much DNA is shared in common. So the two most closely related species on this cladogram are the chimpanzee and the bonobo. So I'm gonna, oh, that's not wide enough. Let's make that a little more obvious. So this right here. So now what I've done here is I've highlighted some species and their common ancestor. Okay, so remember that a clade is a single ancestor and all of its descendants. So we have a single ancestor and everything that is descended from that ancestor. So what I've just highlighted in purple here is a clade. That is an example of a clade. So this clade contains chimpanzees, bonobos, and their common ancestor, which is down here. That's the last time in genetic evidence we have that there was one species here instead of two. Okay, and a speciation event occurred and two species evolved from one species. We can make a pretty good guess in terms of when that speciation event occurred in time. It was about 3 million years ago for these two. And that's based on two things. One, it's based on fossil record. We don't have fossil record for everything, so it's not always based on fossil record. But in this case, we have some fossil record to show where this split occurs. And we can also make a very good educated guess just simply based on the DNA itself, looking at how many differences there are between the two's DNA. We have a general idea of how many mutations happen per unit time. And so if we know how many mutations happen per unit time and we look at the number of mutations that are different between the two of them, we can make a pretty good guess as to how long ago they were the same species. And so a lot of this evidence in terms of timeline is based on that. So there's an example of a clade, chimpanzees, bonobos. You can also look at the clade that includes chimpanzees, bonobos, and us. Okay, so that's a slightly bigger clade. It's this clade right here. So now, again, we're looking at a group of species and their common ancestor. Okay, so that's also a clade. So our common ancestor, when we shared an ancestor with bonobos and chimpanzees, was about 6 million years ago. Okay, and that's when that speciation event occurred back here, where our common ancestor and the common ancestor from chimpanzees and bonobos split off. This is more related to genetic evidence than it is to fossil record. So we don't have as clear of a fossil record here, but we do have good genetic evidence to suggest that this happened. So this is our modern understanding of where all modern species came from, and there's a lot of evidence for this. I'm only like scratching the surface. In the evolution unit, we're going to get into a lot deeper into the evidence behind this, why we know that this is true. So I'm just scratching the surface here, but the genetic evidence is a big part of it. Really interesting. If you've ever had occasion to see Darwin's journal, there's a page from it right there. This is the first time that somebody has ever written down a phylogenetic tree. So Darwin did some really cool scientist did some touring of the Galapagos, looked at a whole bunch of different species, and then based on what he saw in terms of differences of species, decided, I think these things are related to each other, and they seem to be related to each other in a pattern that you can trace back their relatedness in a tree format. Okay, that's a phylogenetic tree. And there's the first one right there from Darwin's journal, which I think is really cool. I like that he wrote on top of it, I think. So he like drew the tree and he was like, I feel like this goes together like this. I mean, he obviously didn't know. He didn't have all the modern evidence that we have for evolution, but good guess. Good guess, Darwin. Anyway, we found a lot of evidence since then to confirm that. So everybody's sort of comfortable here with this idea, the phylogenetic tree. We're grouping things together based on their relatedness. The closer you are on the tree, the closer you're related. Later on, I'm going to get you guys to do a little bit of practice with this. It like walks you through a cladogram in the textbook and gets you to do answer some questions about what is and is not a clade, etc. Nobody's like freaking out at this point. It's just like Maybe no one would type that in the chat anyway. Feel free to email me if that's how you feel. I'm going to figure out a polling system. I was trying to watch some videos over the break about polling systems. Okay, I'm going to keep going. I think after you do the tutorial, if you're where it really does in detail go over like how to make a cladogram, what all this stuff means. If you're after that, if you're like, oh man, I don't get this, then we should definitely talk. Interesting question. So what you're asking is, are the species at the tips of this cladogram, at the tips of this phylogenetic tree, are they continuing to evolve? And short answer to that question is yes, they are. Evolution is still acting on all of these species. Is it possible that the bonobo will evolve to be more human-like over time? Sure. If the evolutionary pressure on the bonobo pushes it in that direction. This may not be a satisfactory answer at this point, but we are going to discuss these mechanisms in a lot of detail during the evolution unit. So I don't want to go super off course here, but it is possible that they could evolve to be more like humans in the future. That's called convergent evolution, where two separate species actually evolve to be more like each other over time. But there's obviously the opposite is possible as well. Divergent evolution could occur here, and different evolutionary forces are acting on the bonobo, and maybe they'll be less like humans in the future. So the short answer to your question is they can become more human-like. They are never going to become humans. So a lot of the differences in our DNA are based on random mutation, and they were selected for by the environment. Again, this we go into more detail during the evolution unit. But the random changes that occurred in the human genome are not magically going to be exactly the same in the bonobo genome, so that eventually they become exactly the same as us. That's I shouldn't say that it's impossible. It is possible for that to happen. However, it is insanely improbable for that to happen, because essentially we would be expecting the exact same random events to happen to the bonobo's DNA, and then the exact same evolutionary pressures, selective pressures, acting on the bonobo, pushing them towards becoming human. It's unlikely, and I say that as a huge understatement. It's tremendously unlikely that they would do that. Now, could they gain more human traits over time as they evolve? Yeah, probably. Are we currently pushing the bonobos to extinction? so that we will never find out the answer to that question? Yes, unfortunately. That's sad. Hopefully not. Okay. Let me answer that question for you, Datina. Yes, the change is random. In other words, the differences that build up between two separate species is random. What parts of their DNA are going to experience a random mutation is random. However, whether or not that mutation that happens is selected for, that is, it becomes more common in the population and the species evolves to become like that, is dependent on the environment. So we are going off course here, by the way. This is all for the evolution unit. But okay, let's say that you've got an animal like a bird, okay? And due to a random mutation, it gains webbed feet. If that bird is living in a tree, okay, that's its habitat, then webbed feet are not a positive mutation for the bird. Okay, it doesn't help the bird at all. And so that mutation would be selected against. And so it would be less, in other words, it's not going to get passed on. And so that mutation is not going to become common in the population because the offspring of that bird are not going to be more likely to survive. They're probably going to be less likely to survive. And so that mutation would die out. But if that bird were to be an aquatic bird and it gains webbed feet through random mutation, then that would be selected for. It would help it swim. And so you would see in a future generation, more members of the population are going to have that trait until potentially the entire population has that trait. Okay, that would mean it's being selected for by the environment. So when you say adaptation, the random mutations that are occurring can be adaptive. They can cause better survival for a species, or they can be maladaptive. They cause worse survival for a particular species. And the environment decides whether or not they are selected for or against. So that is what natural selection is. The environment is doing the selecting. That's correct. Yep. What you're describing is natural selection. Hopefully that answered your question. We will go into this in a great deal more detail in the evolution unit. And I lost my screen here. What the heck? Sorry, guys. Reset my Wi-Fi here. I know that. It stopped broadcasting for some reason. I'm on the horse screen. Come on. Every once in a while, my iPad decides that it's just done broadcasting. No, still not going to work. Come on. We can reset this. Working perfectly 30 seconds ago. Why has it given up? I may have to restart my mirroring software. Sorry, guys. Technical problems of teaching online. Okay, I'm going to restart the mirroring software. Let's see if that helps. What the heck? Airplay on. Sorry, guys. Bummer. It's just loading. Loading. Nothing happened. It's strange. Okay, I'm going to try something else here. I'm going to restart my iPad real quick. Hopefully that doesn't take too long. This one's pretty good at restarting fast. Should be okay. Give me 2 shakes, guys. Sorry. Come on. Hey, there we go. That's what it took. All right. Okay. Sorry, we got a little bit sidetracked there. So I wanted to show you a cladogram here for all of the phylums of animals. Okay. So this is just like this cladogram here. The only difference here is this one doesn't actually give a date for when these branched off. You can find cladograms like this that do have the dates for the animal groupings, but you'll notice that they have specific genera that they've used to make these groups. These are the original taxonomic groups. They're not based on genetics. However, the things that we use to classify them genetically also, very luckily, happen to be the major genetic turning points for these organisms as well. So we've done an okay job here for categorizing a lot of these. In other words, once we've used phylogeny, genetic evidence, to check our original taxonomy for animals, we've noticed that it's largely the same. So we did a good job, I guess, taxonomically here, using morphology. It more or less reflects the evolutionary history of these organisms. But you can see how for example, the chordates and the echinoderms are starfish, are the two most closely related groupings here in the animal kingdom. And that might seem very strange, but we actually share the most homologs between us and echinoderms. And if you look back at the... Why am I not able to write on the screen? There we go. If you look at the homologs between these two, we share the most homologs. So that just means if you were to go back in time, our closest common ancestor is closer. We share a common ancestor with echinoderms before we share a common ancestor with any of these other groupings in the animal kingdom. Okay, some of these, like these lophophorates, we don't exactly know where they fit in the phylogenetic tree. Notice the question mark. What is the common ancestor and where does it fit? We don't have enough genetic evidence to actually place them in a location. So this is mystery. But a lot of these other ones, though, we can tell how closely we are related to them through genetic evidence. And a lot of the, like I mentioned, the morphology actually comes along with that, so it's related. So in other words, let me give an example here. So collimates, or actually body cavity. Here, let's use body cavity as one of these. Okay, so there was a time in history where that was an evolved trait for these organisms. The idea of having a body cavity. In other words, we have body cavities. If you open us up, there is like a space, and the organs are like in the space, in the cavity. The organs are kind of separate from one another in a cavity. That's a body cavity. Not all animals have that. So, for example, flatworms and radiata, which include the cnidarians and the ctenophores, those are things like jellyfish, and porifera, which are sea sponges, they don't have that. They don't have a body cavity. And so at one point we shared a common ancestor that did not have a body cavity, and then that trait evolved. A body cavity evolved. And any of the modern organisms that share that common ancestor, well, we all have a body cavity because we share the homologs from that common ancestor. But because these guys evolved earlier than that, these guys on the end, they branched off and they did not have a body cavity, and they still do not have a body cavity. So, and then that was a random mutation. So all of these are things that arose from random mutations. Okay, anyways, this is just an example of some major taxonomic groupings here based on genera. You can look at this if you like. It's not super important. But there are some significant branches here, Protostomes and Deuterostomes. That's the idea of whether or not they're, when they first develop as an embryo, do they grow their digestive tract from the bottom up, from anus to mouth, or from mouth to anus? And protostomes are the more ancient ones, and they grow from mouth to anus, their digestive tract. And some of them don't grow it all the way, by the way. A lot of flatworms only have the tube going halfway down, and then it just ends. It doesn't have another end. Whereas the deuterostomes have the tube growing from the other end, anus to mouth, and they always have a complete digestive tract. So those are these ones over here, the deuterostomes. Okay, a lot of complex organisms are deuterostomes. And then radial symmetry and bilateral symmetry, that's another big split here. Again, this is indicating genetic relatedness, but it's also incorporating the taxonomy here, the morphology that confirms that. So what I'm going to get you to do, okay, I'm going to stop talking, by the way. What I'm going to get you to do is, really? Making me log in again, eh? Didn't feel like I was gone that long. What I'm going to get you to do is watch this video, this Khan Academy video on phylogenetic trees. It just goes through how to make a tree. It talks a little bit about the terminology again. And it talks a little bit about how to read a phylogenetic tree. Then I'm going to get you to read tutorial 1 on page 22 and 23, and then do practice questions 1 and 2. They're fairly self-explanatory in that they kind of walk you through how to read a phylogenetic tree, and then they ask you questions about it. But the unit quiz has a number of these. I didn't take them straight from this, but they are similar style questions to this that I've put on the final quiz for this unit. And then there's some analysis questions that go along with that. It's on page 25, 3, 5, 6, and 9. Okay, we somehow I intended us for us to finish this during block 1 before the break, which is unlikely. So there's a second component to this, which I'm just going to talk about really quickly now, and I'm going to get you to do them all together. There is enough time. So the second part here is just this idea of kingdoms, okay? And as I mentioned here, the kingdoms are the level right below domain. Okay, so we've got our three domains: archaea, eubacteria or bacteria, and eukaryotes, eukaryota. Okay, those are the three domains, and they branch into 6 kingdoms of life. Okay, those are the 6 main taxonomic groups. What we're going to be doing throughout the rest of the unit is getting into the details of the 6 kingdoms. In other words, you should be able to identify what is a protist. When you see a creature out in nature, you should be able to say, I know what kingdom that creature belongs to based on what we learn about each kingdom. Okay? So what I'm going to get you to do here, this is not super complex here, but you're also going to read page 27 in your text, and it goes over just the very basics of these 6 kingdoms. What makes something a bacteria? What makes something an archaea? What makes something a protist? What makes something a fungi? And then just give me some of the basic characteristics for each. Okay? Huge detail is not required here. In fact, the page 27 does not go into huge detail. But it gives you the basic details on these. Okay? And then just provide 2 examples at the end. I should just cut out again. This thing's really giving me a hard time today. I don't know why. Anyway, 2 examples of each of these. Try and give examples of actual species, okay, for each of these. And you may have to look them up, and that's totally fine. Okay, this is going to set us up for tomorrow, because tomorrow we're actually going to get into detail and start talking about these kingdoms. We're going to start with eubacteria or bacteria, and we're going to go into detail and really flesh out what makes something a bacteria. What are some examples of bacteria? How are bacteria constructed? And then the next day we're going to talk about archaea, and then protists and fungi, and that's sort of the layout for the unit, is that we're just going to be talking about all of these kingdoms. But for today, all I want you to do is just list what are the basic characteristics of each of these kingdoms, and you're going to do that after you're done the phylogenetic tree stuff, which shouldn't take too long. This may take you to the end of learning block 3, though. I will mention that the first few days are a little bit more work than typical. So I've kind of jam-packed these days a little bit. And the reason for that is I'm just trying to make sure we get 2 units done before the end of the first block, before you go to your other class. If I don't do that, what ends up happening is it cuts the 1 unit in half, and then when you get back, you won't remember a lot of the stuff. And then you got to write the quiz and do the project, and it makes it much, much more difficult. At least I know that from previous quads. So I've rearranged a tiny little bit here, and I've squeezed a little bit extra into the first couple of days. It's not quite this busy. So just so you know. Okay, so what are you working on? Watch the video on phylogeny. Try the tutorial, okay, and the practice questions. They're not that complicated, I don't think. Try these analysis questions about phylogenetic trees from the text. And then you're just going to list the basic characteristics for each of the kingdoms. Okay? And some examples. Seems like a lot, but what time is it now? 12 o'clock? You have about 2 hours, so normally we would easily do this in 2 classes. So if you need anything while you're doing any of this, let me know. I'll be here the whole time. I will be in the Google Meet during Learning Block 3 to answer your questions. And if you have questions from the homework problems, I always take them up at the beginning of the next day. I always ask if people would like them taken up at the next day, at the beginning of the next day anyway. So if you feel like your question might apply to somebody else, you may want to save it to ask during that question take-up time at the beginning. Okay, guys. Let me know if you need anything. I'm going to let you get to work on this stuff. Do we have a classroom? Do you mean a physical classroom in the school? Or do you mean like a Google classroom, or what do you mean, Tamara? Tell me more. like a Google Classroom. There's no Google Classroom. I've moved over to Brightspace. So the Brightspace class, which is this right here that you're seeing, that is our quote unquote Google Classroom. That's our online learning environment. That's the one that I use anyway. The board is asking for people to move over to Brightspace. There's a lot more tools available for assessment and stuff like that on Brightspace that they don't have on Google Classroom, and you can integrate stuff from Google Classroom right into it. So it's a pretty good system once you get it going. I think that they're pushing us away from it. I don't think that the board wants to pay for Google Classroom anymore, which is the reason for it. I think that's what's happening. Okay, I'm going to move to my screen here where I put up all this, what you're working on. Yeah, Google Classroom had its benefits. I didn't mind it. I kind of agree with that. And if you guys need anything, just let me know. Okay?